Today, we're going to talk about what happens when genetic information is not copied correctly. In the last lesson, we completed talking about the process of protein synthesis, where DNA, the message is transcribed in the nucleus to make a mRNA molecule. That mRNA leaves the nucleus, heads out in the cytoplasm through the nuclear pore to the ribosome, where the protein is going to be made. Now, what's missing from this picture is over here in translation is the tRNA molecule that brings the amino acid to the ribosome is missing. But this is the part called translation that occurs on the ribosome. So what we want to ask ourselves is what happens if this information is not copied correctly? And before we start, let's make sure we review a word called mutagen. A mutagen is a substance that can cause a change in DNA. So what might cause a mistake in this process of transcription would be something that could be induced by a mutagen. Some examples of mutagens could be carcinogens, like found in cigarette smoke, X-rays, UV rays. So those would be things. So you may ask yourself, well, why doesn't everyone have a mutation? And actually, we do. A lot of mutations exist. And it depends where that mutation occurs and what might happen. So if the mutation occurs in the germline, which would be in your gamete, your sperm and your egg, then that's significant because every cell is going to have that mutation. And those mutations then can be passed on to offspring. If it's a somatic mutation, so it's a mutation that occurs in the body cell, then that's going to have a little effect because it can be confined to that cell and only future daughter cells of that individuals. So those mutations cannot be passed on to their offspring. So, and again, it depends on what is the potential cause of the mutation. If we take a little bit of a closer look at gene mutations, so where's the mistake or how big is the mistake? So a gene mutation focusing on one gene, and there are two types, point mutations and frame shift mutations. If we take a closer look at point mutations, that's a change in just a single nucleotide, an A, T, C, or G. And I apologize, the writing got a little crazy there, but that says A, T, C, and G. So there is nothing that's being deleted or added. It's just kind of like a switcheroo. So if there's a point mutation in which the mutated codon codes for the same amino acid, for example, this codon codes for glutamine. CAG also codes for glutamine, then there would be no effect. So it's considered a silent mutation, and really nothing has gone wrong here except the message is different. Missense is an example when a mutated codon codes for a different amino acid. So CAA is glutamine, but CCA is proline. So that might make a slight difference. We say variable because we're not really sure of what kind of impact it might have. The protein might not fold properly, but in a sense, the protein should still otherwise be intact. Nonsense mutation is when a mutated codon is prematurely stopped. So CAA is supposed to be glutamine, but UAA, if that C mutates into a U, it would stop. This can be serious, especially, it depends on where in the gene. Is it the beginning of the gene or the end of the gene that this actual mistake happens? So those are all point mutations. If we move away from this is if an amino acid, this can have some serious results. If a nitrogen-based nucleotide actually is inserted or deleted. So deleted would mean one is removed, inserted would mean there'd be an extra. So the example I used in class was if you read the sentence in blue, the fat cat ate the rat. If we delete the F and we try to read it again, it becomes unreadable. The message is no longer clear. And the reason we're grouping the letters in 3 is because that's what happens with the nucleotides. They're grouped in letters of 3. So in class, I had the students translate this message right here and translate this message and see what happens when you insert an A. Chromosomal mutations, usually more serious, can result in death, shortened lifespans, or developmental delay. So you can see from, these were all vocabulary words, and they all sounded confusing, but when you look at this picture, it can really help. Deletion, it means entire segments of the chromosome, or, this could be 1,000 genes have been deleted. This could be 1,000 genes duplicated. This can be that the genes, the sequence are turned upside down. Insertion, genes from one chromosome can be inserted into genes of another chromosome. Or translocation, it could kind of be a flip-flop. Not only are they're switched, they're also flipped. Again, serious consequences usually result in death, shortened lifespan, or developmental delay. So tonight for homework, students were supposed to make sure they have finished reading and learning about mutations from the textbook. That was the assignment. And students were also assigned a gene and chromosome mutation worksheet. The first page is merely the same information that's in your note packs. The second page is where there start to be questions and to actually put this into practice. So if you look at number 1, there are several types of gene mutations. List 2. What do they have in common? How are they different? So if you go back to your notes and you go to the page that talks about gene mutations, you're really comparing point mutations and frame mutations. How are they alike? How are they different? So that homework is due tomorrow, and if you need assistance, I will be here to help you.